Hello, everybody. We're going to talk about mitosis today and maybe a little bit of meiosis. We'll see how far we get. You may want a pen and paper today. We're going to do at least one mitosis problem and maybe a couple other notes. And I think I mentioned before, my plan was to have this particular topic be about a lecture and a half. And so that on Wednesday, we would have about half the lecture time would be review. And then Friday would be entirely review for the midterm. Haven't already started, I do encourage you to get started studying for the midterm. I've already received some questions, so keep them coming. And if I have some good questions to look at for the review period, then all the better. It's always a good idea if I know what you guys want me to talk about for the review period. That's always a very helpful thing to have. So before we get into mitosis, let's just a quick review from last day. We were talking about some different definitions. We had chromosomes, sister chromatids, centromeres, and so on. So this one says, what is a centromere? Is a region in which A, centrosomes migrate to during mitosis. The nucleus is located prior to mitosis. C, chromatids remain attached to one another until anaphase. Or D, chromosomes are found in the nucleus. So the correct answer here is this one here, number C. So remember, we were drawing some chromosomes, and so there's my chromosome, and in interphase, it's going to replicate, and it's going to have an identical chromatid, and that there is the centromere. So the centromere is actually a region of DNA, and that region of DNA is where they're linked until they separate during mitosis. I have some more pictures of this I'll show you in a moment. So let's talk a little bit about interphase. You probably remember a little bit about interphase. Interphase was named by mitosis people, so they thought of it as the intermission or interruption, but in fact, most of the cell's life is in interphase, whereas mitosis is the interruption. So if you look at this diagram here, they're showing interphase as a huge part of that life cycle of the cell. So human cell, a typical life cycle is anywhere from 16 to maybe 24 hours. So mitosis is maybe 1 hour of the whole thing. So a very small part of the actual life cycle. So take a look at the interphase. You can see we were broken into 3 parts. So we have G1. So G1, by the way, actually stands for gap. So what do we mean by gap? Meaning one of the gaps between mitosis. Because like I said, people were studying mitosis originally because it was interesting and everything else was something else, an intermission or an interruption or a gap. You're starting to see a lot of textbooks also call this the growth phase. I like that one better. And so this would be gap 1 or growth 1. And what is going on there? Well, pretty much they're growing, right? So they may rest there. Some cells don't necessarily undergo division all the time. Maybe they need to be damaged or something like that first. A lot of our cells do, though, divide all the time, like our skin cells are replicating repeatedly, whereas our liver cells only do it when they're damaged. So like I said, mostly we're talking about growth there. The S phase, as you can see here, S stands for synthesis, and that's where the DNA is replicated. I don't know why we don't call it the R phase, because DNA synthesis is actually called DNA replication. That's the technical term for DNA synthesis, is replication. So remember that word. So G2, you can see, is just another growth or gap phase. We got a little bit more growth going on. Sometimes we can start to see the centrioles a little bit more visible into the G2 phase, but we really want to talk about mitosis. So what is mitosis? You can see the definition of mitosis here. It says really we're talking about division of the nucleus. So we're really looking at just the genetic material. Cytokinesis, so there's that word again, cyte, right, like cytoplasm, is the division of the cytoplasm. So usually these things are happening somewhat simultaneously, not always necessarily the case. So let's take a look at mitosis, and maybe you remember the phases, maybe you don't. I'll show you a word game that I used when I was in high school. I learned this from my teacher. And she said, remember this little thing here. I think this is called a mnemonic. Play me a tune. So that means we have prophase, metaphase, anaphase, telophase. So that's how I remember what order they're in. But most of the textbooks are starting to put in a little something in between. So sometimes I've seen it called late prophase. Sometimes I've seen it called early metaphase. But our textbook is calling it prometaphase. So a new one to learn. Yes, that's up the end of my screen here. So let me just squeeze it in somewhere here. Let me see if I can erase that. Apparently not. Oh, there we go. Now that worked. Okay, so prometaphase. There we go. So one more new one to learn, and I'll talk about what's going on at the different stages. You're probably somewhat familiar with this. Most people have seen it at least. You at least cover it in, I think, grade 9 and grade 11. So there are the stages. The previous one was an animal cell. This is a plant cell. You can see they're listed there: prophase, prometaphase, metaphase, anaphase, telophase, and cytokinesis, which kind of usually happens roughly the same time as telophase. Sometimes late anaphase, it gets started. Again, it kind of depends on the cell type when cytokinesis is happening. Sometimes it happens afterwards. Sometimes it kind of happens simultaneously. Even in one species, different cells have a little bit different quirks in terms of how they do their cell division. So let's take a look at the stages. We've talked about interphase already. So one of the things to remember about interphase is that we've already had the DNA replication. Okay, so that's important to remember because, you know, when we're thinking about the human chromosomes, right, you probably know that humans have 23 pairs of chromosomes. So that's 46 chromosomes total. So technically at this phase, since things have replicated, every chromosome, so here's my little chromosome, we'll draw one for you up in the corner here, has replicated and now has a sister chromatid. So if you really want to count, you could think of it as almost 96 chromosomes, right? Or 46 sets of sister chromatids. So remember that terminology, right? We have these terminology, chromatids, okay? That's an important word to know. So why don't you take a look at this diagram here and see that we have different parts of the cell are labeled, okay, and with fluorescent diagrams. So the DNA, for example, is blue. The microtubules, so microtubules, these are actually spindle fibers, okay? That's just another name for spindle fibers is microtubules. They're in green, and the intermediate filaments are in red. So let's take a look at mitosis. So here's prophase. So in prophase, we have a couple of things happening here, right? We have these here are our centrosomes, and they start sending out the spindle fibers. They start sending out the microtubules. Other thing that happens is that the chromosomes condense. So I can write that down if you want. We have formation, sorry, this is going to be really small, of the spindle fibers. Really messy today. I don't know why I can't write very neat. Apologize for that. And the other thing is they start to condense. So maybe I'll put number 2 down there at the bottom. We have the chromosomes condense. Okay, so this is important. This is why we start to distinguish prophase from prometaphase. Okay, because you're going to see really this has to happen first before you get to the next step. So prometaphase, things are condensed. Okay, that means they're easy to identify the different chromosomes, and the spindle fibers are going to start to invade the nuclear space. So how they're going to invade the nuclear space, we're going to have the disappearance of the nuclear membrane. So that's 3 things in this case. So I will write those down. We'll start off at the top somewhere. So we have nuclear membrane dissolves. So remember those nuclear lamina, nuclear membrane dissolves. Remember those nuclear lamina, they are going to basically disassemble to allow that to happen. Okay, so nuclear membrane dissolves. Number 2, we have the microtubules or the spindle fibers. So I'm just going to use MT, start attaching. And where do they attach? So remember that word, they attach to the centromeres, sorry. Get my words mixed up. Okay, and what did I say what the third thing was? I don't know if I said the third thing, but there is a third thing. So notice that the centrosomes are opposite. So they're at the poles of the cell. One is at the far end and one is at the other far end. And in this diagram, you can see one is at the top and one is at the bottom. Okay, so what happens at metaphase? So everything is attached. So that means that all of the chromosomes are attached to all the spindle fibers. And so basically what's happening at metaphase, they all line up. So metaphase, really just one thing to say about this. We have sister chromatids are lined up in the middle. So the middle has a name. We call it the metaphasic plate. And I like metaphase because M for metaphase, M for middle, helps me remember what is going on. Okay, anaphase. Anaphase, A is for action. And so now we're basically having the separation. So separation of what? Separation of the sister chromatids. So these are good things to know because, you know, your midterm is coming up and they're asking you in a short answer question, what is happening in anaphase? Okay, you got to tell me what's separating. Don't just say separation. Don't just say separation of the chromosomes. Technically, they're sister chromatids. Okay, so you got to have the terminology correct in this kind of case. Another thing you can add on here, what is happening in anaphase is we've got some cell elongation. So, and that's important because eventually we're going to separate this thing into 2 daughter cells. You can see the nice diagram here, that electron micrograph, really nice colors in there. Not really sure what's going on with those intermediate filaments. They look like a little bit of a storm, but very cool looking. So telophase, everything's separated. The job is done. We just got to clean up. So we have to reform our nuclear membranes. So we can say that in nuclear membranes reform. And often at this stage, it doesn't necessarily happen right away. Again, depends on the cell type, is the chromosomes become less dense. So they, you know, kind of loosen up, I guess. I'm not sure if there's a term for that, but they become less dense. So that is mitosis. Hopefully there are no surprises there. Most people have seen mitosis somewhere before. I've got a video here I will play for you. Here goes. And we can watch mitosis happen in an animal cell. So if you take a look at this mitosis here, a lot of this, it's basically prophase. It's already condensed. Metaphase is very slow. And you'll see once metaphase is done, anaphase happens very, very quickly. It's very cool. Just watch for it. They're lining up, trying to figure out, you know, where everyone's place is, it looks like. Ready for it? Get ready for it. Almost there. Like I said, metaphase takes a while, and then there we go. There's anaphase. Very, very quick. So there's a few of these things on YouTube. If you're interested in mitosis, meiosis, check them out. And you see the last stage there, we have some cytokinesis happening. So the cells are dividing their cytoplasm. Okay, very nice video. So let's talk a little bit about these spindle fibers. Okay, remember that spindle fibers are basically microtubules. That's a good thing to know because those words are used relatively interchangeably. And something else to know, you can see that the centrosome is sometimes called an aster. An aster kind of means star. And so sometimes I've seen that terminology used, not too often, but once in a while. And they are pushing and pulling. right? So some of those spindle fibers push, some of them pull, and the whole goal there is to separate the two sister chromatids once everything is lined up. This here is your video as well. I will play this for you, and you can see, I guess it's not going to play for me today. So sorry about that. I guess next time I'll play it. So the one thing that I mentioned last day is what is going on at the centromere. And the centromere has these proteins here, kinetochores. So right in the name of that, kinete, right? Kinetic. That has something to do with motion. So these are motor proteins. And you can think of these as almost like little winches. They're little motor proteins, and they're found on the chromosome, and they're just going to climb along that microtubule, and once things are ready to separate, they're going to climb along the microtubule and bring that sister chromatid to the pole. Okay, so there's another promise for you. I'll definitely have some question about motor proteins on the midterm, so you can take a note of that. So here's another nice little picture. I kind of want to talk about cytokinesis, but I want to show you this picture first because what we have here is on the left, they've stained microfilaments. So this is microfilaments. So remember, microfilaments are also called actin filaments, made out of actin. And we have microtubules stained over here, microtubules stained over here. So the microtubules are the spindle fibers, and the actin filaments, remember I had mentioned that they were kind of near the membrane, just sort of under the surface, inside the cytoplasm. And so microfilaments are used for the last stage here, which is the division of the cytoplasm. So in animal cells, we have this pinching that happens here, and that is all done by the actin filaments. And so the cell pinches, and that thing is called a cleavage furrow. So I have some pictures here of some cleavage furrows, and you can see it's kind of like, I think of it as like a little lasso or a little, like the strings you'd have on a hoodie or something like that. And they basically constrict the membrane until it gets nice and tight, and then you end up with two cells that separate from one another. There's some more pictures. We've got some, looks like some sea urchins, and the ones on the bottom are some cancer cells of some sort. So what if you have a cell wall, like a plant? You can't do that. Plant cells have rigid cell walls, so they do things a little bit differently. What they do is they have cellulose for cell wall, hopefully you know that, and they have proteins that build those cell walls. And so what happens is those proteins are made at the rough ER. These are membrane proteins. And at the rough ER, they go to the Golgi, and the Golgi sends them to something called a cell plate. So a cell plate is right here in the diagram. You can see all these little vesicles. Basically, they line up, and eventually they merge, and eventually they form two new membranes, one for each daughter cell. And those are enzymes that make new cell wall material. So new cell wall material is eventually made and filled into the gap in there. Okay, so there's a diagram of a cell plate, or not a diagram, sorry, an electron micrograph of cell plate. I think I showed you a similar picture to this before, maybe for lab 2 if I remember correctly. And you can just make out the cell plate there where the two cells are just about to finish dividing. So there's some more pictures of the plant cells. You can see all the stages there. I found this diagram, and I thought I'd share it with you for a couple of reasons. One, the spelling is atrocious. Holy cow, look at this. Interphase, later Prophase, a couple of things like that. But I want to also point out that they are actually labeling a stage in between prophase and metaphase. So that would be Prometaphase that we're talking about there. And they can't keep the font size consistent. So, you know, enough little interesting things on this diagram. Thought you might find that amusing. Okay, so I'm going to show you this video. This is one of these 3-minute videos, and then we have a mitosis problem that I want to show you. You're going to have a mitosis-meiosis problem on the midterm. So the first part is understanding how to do the mitosis part. But let's watch the video first. If I can get the video to play, I'd be very happy. Video is not playing. let me just see here. I think I have another version of it. I think I have, let me see here. Just give me a moment. I'm pretty sure I have the video somewhere. Just have to go find it. Okay, not there, not there. This is what happens. You get a new computer and then you can't find all your videos and stuff. Okay, I'm pretty sure this is it. Oh, my computer's thinking. I see what's going on. Okay, so the video was kind of stuck in the cloud. It wasn't downloaded on the computer, but it should just take about 15 seconds here to download, and then we can watch the video. Sorry about that. About halfway there. Stuck at halfway, but okay, no, it's going in. I know the suspense is killing you. Okay, 80% done, almost there. I'll play this for you right away, and you can enjoy the video. This is what I get for being on the Wi-Fi today instead of the hardwired internet. Okay, so I just got to share that with you. Okay, where did it go? Okay, so this is fun. My computer is not happy with that video. Let me just see here. I think I found somebody had it uploaded on YouTube. So let's try that. Unavailable. Okay, so I'm really sorry about that. But at least it's just mitosis, and usually most students are relatively familiar with mitosis. So sorry about that. Hopefully I will have the one for meiosis working when we get there. But it looks like it's invalid. So I just got to figure out how to, I think it's not happy with me here at the moment. Oh, I see there's some questions here. Hopefully I didn't miss anything too important. Yeah, so hold on, I'm just going to go back to the PowerPoint. No, that's not the PowerPoint. That's the PowerPoint. Okay, so sorry about the video not working. Technical difficulties, not unusual for this year. So here's a nice little quiz thing we can try. So it says, put these in order, label the phases, okay? So, and then what are X and Y? Let's start off with X and Y. Hopefully that is easy, okay? So X is centrosome. Centrosome. Remember, they were also calling it the aster, and Y is the spindle fibers. Okay, so that's easy. Now let's put these in order. Okay, so the easiest thing to do is think, okay, where I see two cells, that's probably later in mitosis. So I'm just going to ignore that at the moment. You know, I guess we could label F, and F is kind of when everything is done. So we'll call this after telophase, or maybe telophase might be the correct answer because you can see the nuclear membranes have reformed. Okay. So that's good. So let's take a look at this. We've got some where the cells have replicated their DNA and some that they haven't. So I'm looking here at D. So D, that's number 1. And I'm guessing we're going to call this interphase. The reason why I'm not 100% sure about that, because in interphase, the chromosomes are not going to be condensed, but I think just for being simple, that's why they've condensed them, so you can count them and see them easily. So interphase, those chromosomes are going to be replicated, and uh-oh, what happened to my computer screen here? think the video just popped up for me, but it says error. So hopefully I'm still sharing this PowerPoint with you because I just clicked back to it. Okay, so then they get replicated, and then we're looking at A. Oh, come on. Not a fun day for technical difficulties. Okay, so they're replicated, they're condensed. You can see the nuclear membrane is starting to disappear, but it hasn't disappeared yet, so that's definitely prophase. So what else is going on here? You can see after that, they are lining up. So here we go, this is 3, and this is going to be metaphase. They start to separate, so that's number 4, that's anaphase. And then they're basically separated, so maybe this is telophase. Like I said, we'll call this late telophase, maybe F. Okay, so make sure you take a look and understand what's going on in each of the steps. There'll definitely be a couple of questions on the midterm with that. The big one are these, what I call these mitosis, meiosis questions. Like I said, it's going to be, it's going to be worth 5 marks. 2 out of the 5 marks are going to be on the mitosis. So we're going to try this one right now. And I'm hoping I have enough, maybe I don't have quite enough room on the screen. So maybe what I will do is I will switch to a whiteboard to do this question and show you what is going on. And so let me just switch to the whiteboard for you. There we go. So we're talking about mitosis, right? So mitosis, and it told you that you have a cell of n equals 3. So we haven't really talked about these n numbers and all that stuff too much, I thought, all this semester, but hopefully you remember what that means. So this is the haploid number. Okay, so it's good to know if you're a diploid organism, then we want 2n equals 6, okay? So 2n is the diploid number. Okay, so what did the question ask us for? The question asked us for interphase, metaphase, and the daughter cells, okay? So here we go with interphase. So what you're going to do for this, and the question will ask you basically to draw the cells, the DNA, and the spindle fibers. So you don't need to draw anything else, okay? So interphase, metaphase. Okay, maybe I will make that a little larger in a moment. And we have the daughter cells. So you can get fancy if you want, but what I really want to see is just that you can count, okay? So in this case here, you get half a mark. The question would probably say something about a cell having n equals 3, how many chromosomes are going to be in the diploid cell, and you're going to draw that for me. So if the answer is 6, you can draw 6 chromosomes. So just like this, 1, 2, 3, 4, 5, 6. And that's it. You get half a mark just to show me that you can count and then give me the right number, 6. Okay, so the next one, we're going to do metaphase. So metaphase, remember, these things are going to line up around the center. They are also going to be replicated. So remember, they're going to look a little fatter. They're going to have a sister chromatid, okay? So again, you don't need to be really fancy with your artwork. You can just, if you want to make them all fancy, you can like this, okay? I'm making mine, first one at least, fancy. So you can see there we go. We've got 1 chromosome, 2 sister chromatids. The rest of them I'm just going to draw like this. It's a little bit easier. And they're lined up along the middle. So notice I am going to make 6 of them, right? So there we go. And I didn't leave myself a lot of room, but I was able to just cram them in. So what I said, like I want you to do, is you're going to draw the chromosomes and the spindle fibers. So the spindle fibers are going to go to the end somewhere. So wherever you think there might be a centrosome. So there we go. There's spindle fibers going in one direction, spindle fibers going in the other direction. Okay? So that's what it's going to look like. So I'm looking that you've not changed the numbers. We still have 6 chromosomes. I'm looking to see that they are replicated, and that we have the chromatids and the spindle fibers in the right place. If you want, you can do it the other way, right? You can have chromosomes in the middle like this and spindle fibers off to the side. It doesn't really matter. Both are fine. So the last thing is the daughter cells. So I could draw anaphase if I wanted to, and I'll do that when we look at meiosis. But the daughter cells, it's just a matter of, okay, so half of those spindle fibers are going to the top cell, half of them are going to the bottom cell. So you're going to have 6 chromosomes again. 1, 2, 3, 4, 5, 6. 1, 2, 3, 4, 5, 6. If you want to draw these nicely, you can make them a little thicker. I see some people are having a hard time with their connection today, so it's not just me. If you want to draw them thicker, that's fine, but mostly in this case, I'm looking to see that you've counted, okay? So keep talking about people not being able to count, because there's always people who mess this up. Somewhere in here, suddenly there's 12 or 3, or I get weird numbers, right? So this question is just a very basic one, and I'm showing you that we start off with 6, and we finish with 6, okay? There's a few things that obviously happen in the middle. I could get you to draw anaphase instead, but like I said, there's 2 things you need to draw. The chromosomes and the spindle fibers, and that's what these questions are all about. So we will see some more practice questions, by the way. And like I said, I want to kind of get to meiosis first, and then show you what a full question actually looks like. Okay, so we're going to move on, and we're going to talk a little bit about a couple other things around mitosis, and we're going to talk a little bit about meiosis today. So one other thing to consider with mitosis is that the cell cycle has to be regulated. Okay, what do we mean by regulated? It means we want to turn it on when we need it and turn it off when we don't. And it kind of just depends on the type of cell. So skin cells, they divide continuously. And that makes sense. Our skin gets damaged, it flakes off, we need to replace our epidermal layers. Other tissues, only when triggered, often by damage or something like that. So when I need more liver cells, I can make them. But other than that, I don't want my liver to keep, I don't want those cells to keep dividing and growing. I don't want a giant liver. I want a good, normal sized liver. Some cells don't divide at all. They just mature, and that's kind of the end of them. And that's true of a lot of your neurons. Once they have matured and differentiated, that is the end, and they don't undergo mitosis. So lots of different scenarios. So let's take a look at that cell cycle again. You can see I have G1 and the S phase and the G2 and the mitosis phase in there. And there's a few things to take a look at about what is going on here. So I actually, it was a little bit out of order, but I'll just click on them all at the same time. And so let's take a look at the G1 checkpoint. Okay, so as I mentioned before, some cells will rest for a while in G1. And if you're a neuron, you may rest forever. And so they're going to grow and they're going to stop. And before they proceed, you don't want to start synthesizing your DNA and proceed and then find out that you've wasted all that energy. So you're only going to do it when you're checking for things like the cell size, nutrients, growth factors. I'll talk about growth factors in a moment, or DNA damage. So you're looking at all those kind of things. There's also a checkpoint at G2. Looking for cell size, that the DNA has been replicated, and there's a checkpoint at anaphase to make sure that the chromosomes have actually fully attached. So I was going to write a note for you. I'm going to go back to the whiteboard, and here's our whiteboard. Maybe I will just go down, make some space down here, and I kind of had a list for you of things that are involved in regulation of cell division. So we'll call that regulation of cell division. So not a huge note. I'm going to divide this into 2 categories. So external cues. So what do I mean by an external cue? I mean something outside of the cell, a signal from somewhere outside of the cell. And internal cues. So you can see this is not going to be a big note. There is more information about this in the textbook if you want to read more detail into this, but I'm just keeping this simple this semester. So what external cues are we talking about? So is the cell attached to something? Okay, sorry, I'm just going to shrink this just a tiny bit to make sure I can fit everything in. This is called cell adhesion. Cell adhesion. So why is that important, right? Why does the cell have to be attached to something? Because this is actually the normal state of human cells. Most of our cells are attached to something else, right? We've got connective tissue attached to bone, you've got tendons and the ligaments, and all these things are attached. And if they're not attached, you can't have a cell floating around your body just growing and dividing willy-nilly, right? That would be bad. That would be cancer, and that's not good. So it has to be attached to something. Is there room? So again, a cell isn't going to go and try to grow when there's a neighboring cell in the way. So this is sort of a check for damage or growth, right? So we call this cell density. They don't want to get too thick. They want to make sure things are just right. And then the last thing I mentioned are growth factors. So growth factors include things like hormones. So this is something that's important, you know, when we're growing, for example. You start off as a juvenile and you grow and different stages in your life. People have growth spurts, puberty and things like that. And these growth factors are telling cells, hey, time to grow. There's other growth factors that when we have a scratch or a wound, the growth factors get released and they tell cells nearby start to, you know, do some repair and you know, fix things. So there's lots of signals around growth factors. I'm not going to get into them in a lot of detail because it just takes way too long. But if you take some more cell biology or physiology classes, you will talk about growth factors. Okay, so what are these internal cues? Okay, so we've already talked about some of these things like cell size or growth. It's got to be big enough if you want to replicate your DNA. Cell size or growth. Is the DNA replicated? DNA replicated? The question mark. And the other one I mentioned, which is really only regarding certain growth stages, is the state of the spindles. So like in anaphase, are they all attached to the sister chromatids? So like I said, I'm trying to just keep this simple. We could spend an entire lecture on this and it gets really complicated. You want to take a look in the textbook on it, but these are kind of the things that I want you to remember, is that there's signals inside the cell that say, hey, it's time to grow, it's time to divide. And there's signals outside the cell that say, yep, okay, it's time to divide or it's not time to divide. So keep those in mind. Okay, I want to go back to the PowerPoint now and kind of just talk for a minute about cancer. There's an entire chapter on cancer. I can't remember, it's like chapter 47 or something like that you can take a look at. I just want to kind of talk about it briefly. If you take biology, what is it, 200 or 201 is eukaryotic cell biology. There's a whole unit on cancer. It's very complex stuff, but from a very simple perspective, it's cell division that's gone wrong. So cells are dividing and they're forming a mass that is not normal, and we usually call that mass a tumor. So some tumors are benign. Benign means harmless. Some tumors are malignant, meaning they're not harmless and they're causing damage. And usually when the tumors are malignant, we call it cancer, meaning it's causing damage to your body and causing ill health effects. So there's an X-ray of a tumor right there. So something to think about with tumors, which is kind of interesting, is that a lot of the drugs that we have that treat cancer actually target the phases of the cell cycle. So this is talking about how there's some of these drugs are microtubule inhibitors. So cancer cells divide really quickly. If you have microtubule inhibitors, then they're going to slow down those cancerous cells. They might kill other cells, which is why people lose their hair, but the cancer cells are going to be extra susceptible to it. Anti-metabolites, so kind of inhibiting cell replication, again, there's some pretty extreme side effects, but the cancer cells are extra susceptible to that as well. So there's some words for you. You may have seen some of these words. I'm not going to ask you about these on a midterm, but I mentioned benign tumors, malignant tumors, and sometimes you may have heard of people with cancer and it gets metastatic. So what does that mean? That means that it's spreading, right? And that's when it really gets dangerous. People might have, let's say some sort of spleen cancer or liver cancer or something like that, and it's slow and they seem to be doing okay, but the real risky part is when it starts to spread to other tissues. And of course, some tissues are going to be quite dangerous if it gets in the heart or the brain or forms a blood clot or something like that can be fatal to somebody with cancer. So one other thing about cancer, what is cancer caused by? Caused by carcinogens. What is a carcinogen is a question for you. So a carcinogen is an agent that can cause cancer. And probably most people think of certain chemicals as carcinogens, but carcinogens can also include radiation, like ultraviolet radiation, and there's actually some viruses that can cause cancer. How do they cause cancer? They're directly affecting the DNA in some form or another. So UV radiation, for example, damages DNA directly. And if you damage genes that are involved in the cell cycle regulation, then this can lead to cancerous cells. Okay, so unfortunately, we can't cover any more about cancer. Like I said, it just takes a lot of time, and but it's just worth touching on because it is related to the cell cycle. So we're going to spend a few minutes now talking about sexual reproduction and meiosis. And so again, some definitions for you. What is asexual reproduction? This is where a cell is usually in a eukaryotic organism undergoing mitosis. So some of our cells do this all the time. Like I said, our skin cells, you can see there's a paramecium undergoing asexual reproduction, and you're ending up with two identical daughter cells. So what do we mean by sex? This is where we have an exchange of DNA. It doesn't necessarily involve reproduction. So you may have remembered when we were talking about the paramecium is that parameciums have a macronucleus and a micronucleus. So the macronucleus is involved in the day-to-day cell stuff, right? So producing enzymes and whatnot for the cell to live. The micronucleus is for conjugation. So these two paramecium are having a romantic moment, and they are exchanging their micronuclei. It means they're exchanging DNA, but there's no reproduction in this part of the process. It's just, they're just exchanging DNA. So what is sexual reproduction then? Sexual reproduction is what mammals do to reproduce. And so there's some DNA that is going to be shared, but in order to produce those gametic cells, we have to think about meiosis. So I want to show you some of these life cycles here, and we are mostly just going to be thinking about animals. You're going to talk about, if you take Biology 108, you're going to talk about a little bit about plants and fungi, and there's some weird things that go on over there. But animals, most of us understand because we are animals, and most of us take a little bit more interest in what's going on there. So if you take a look, we're going to start right here. Okay, we all started off as one cell, a fertilized egg, and that egg was diploid, so 2N. And so we grew, and we grew by mitosis. So those cells divided, and they divided again. Some of them differentiated, and we had all sorts of cell types. So this is how we grow our body, and we end up as a diploid multicellular organism, right? So that's great. What about when we want to reproduce? So some of our cells undergo meiosis. So they go from a 2N to an N. So they go from diploid to haploid. So if you are a woman, that would be an egg cell. If you're a man, that would be a sperm cell. And so you can see we have a haploid cell here from one individual and a haploid cell from another individual, and then that would be what fertilization is. So hopefully that's pretty straightforward, and people probably have a decent understanding of that. But it's just worth it, like I said, to keep track of all the N's and 2N's, and the counting is where people are going to get tripped up, believe it or not. So wait until we see what a mitosis question looks like, which I think we'll probably run out of time today too to get there. So plants, a little bit different. Some plants can have an entire generation of diploid organisms, and some of them can be tetraploid. There's lots of weird things going on there, but you can see that you've got some of the basic same ideas. You've got meiosis producing gametic cells. So in plants, you've got ovum and pollen, which are basically eggs and sperm, and they fertilize and make diploid organisms. So like I said, not really going to talk about plants too much. They're a lot more complex. And then, you know, if you look at fungi and protists, there are all sorts of weird things going on there. Some of them are haploid, some of them are diploid, some of them reproduce by asexually, some of them don't. So it's not even worth it to talk about them unless you're taking an entire class on fungi or protists. So let's go back to our chromosome terminology here. And usually at this point in class, I have a little box, and there's a surprise in there that I show people. And this is what I pull out. I say, what is this? I said, let's pretend this is a chromosome, okay? And so it's kind of easy to picture that. You've got a bunch of mints in there. And what's going to happen during interphase is you're going to have replication. And so your DNA is going to duplicate. So now I have a copy of my chromosome, and together they are called sister chromatids. So at the center is a centromere region where they're going to join, and they're going to be held together by those kinetochore proteins. So at that point, I'm like, okay, so everyone usually understands that. You've got a chromosome, it's replicated, and now you have sister chromatids. So let me show you this, this picture from the textbook, and then I'll come back to the candy in a minute. I know everyone loves candy, especially this time of year with Halloween coming up. So there's a cell there, and you can see it has 6 chromosomes. So half of those chromosomes, 3 of them are from mom and 3 of them are from dad. So you can see the maternal ones are red and the paternal ones are blue. So 2n equals 6. That means 6 chromosomes total and 3 from each parent. So there's our sister chromatids. We just talked about those, duplicated. There's our centromere. And so what are these things here? So here's some new terminology for you, right? We were talking about sister chromatids, and now we have homologous chromosomes. So what does that mean? We've got chromosomes that are the same, but they're not the same, right? We got one from mom and one from dad. So they're going to have the same genes, but the genes might have different alleles. So maybe from dad, you got brown eye genes. Maybe from mom, you got blue eye genes, or the other way around, right? So homologous chromosomes are something we want to talk about. So they are also going to have chromatids, but they're going to be non-sister chromatids because they're not attached together. So back to my candy. At this point, I pull out of my box and I say, well, what about this? So, and people are looking and they're like, it's a Mentos, but it's not the same Mentos. So like I said, you can think of this as a homologous chromosome. Each of those are replicated. They got sister chromatids, and together they're homologous chromosomes, one paternal, one maternal. And then, of course, you know, people are thinking, you know, what else is in the box? And then I pull out the mini Mentos, which took me a long time to find, but I found some. You don't want to eat them. They've been in my box for a few years now, and I found some different colors. And so what do we have here? We have a different set of homologous chromosomes, right? So maybe the ones on the left are chromosome number 5, and the ones on the right are chromosome number 20. So I want you to think about these diagrams and think about these chromosomes when we look at meiosis, which we're not going to cover today, but I'm going to overview meiosis here. So I'll come back to that in a moment. I'm just going to do this overview and then we'll look at the details of meiosis. So here is before meiosis, and we have interphase. So interphase is exactly the same here, right? You have a set of chromosomes. If we're humans, we have 46 chromosomes, and they replicate. And all those chromosomes now have are pairs of sister chromatids. So they're all replicated. So meiosis actually has 2 stages, and we call them meiosis 1 and meiosis 2. So what happens in stage 1 is the homologous chromosomes separate. So you can see the chromatids are still together. Remember, in mitosis, the chromatids separated. Here, the homologous chromosomes separate in meiosis 1. And then what happens is meiosis 2, that's where the sister chromatids separate. So finish up shortly here. Just want to point out a couple things. We start up here with 2 chromosomes, right? So that's 2n equals 2. And down here we finish off with 1n equals 1. Okay, this is very simple when you've got 2 chromosomes equaling 1. This point here, they've duplicated. This point here, they've separated one separation, a second separation. So remember this, if you're going to start off in meiosis, you're going to start off with one number and it's going to half. Okay? Probably many of you will get this. Some of you will miscount. Like I said, sometimes extra chromosomes start appearing when people do these questions. So I'm going to try to emphasize this again and again. Okay, sorry I went over time. I meant to finish a little bit earlier today, or I meant to get a little bit further today. So, but we will have this done and definitely have some review questions for you on Wednesday. And I hope your day is going well, and I will see you then.